everybody. I would like to talk on the topic improving organophosphate-hydrolyzing activities of recombinant human paraoxonase 1 by mutagenesis approach. Before going to discuss something on the relevant topic, we ought to know what is paraoxonase 1 is. So in fact, the human paraoxonase 1 is a calcium-dependent enzyme. It is synthesized in the liver and secreted to the blood circulation. In the blood circulation, it is predominantly found associated with the high-density lipoprotein particles. So it can hydrolyze a variety of substrates like the organophosphates, lactones, carboxyl esters, and the oxidized phospholipids, and therefore it can be a potential drug candidate. However, the crystal structure of this enzyme has not been resolved till the date, and the chimeric PON1 that has been derived from the different mammalian species, that PON1 crystal structure was resolved in the year 2004, which reveals that it is a six-bladed beta propeller having a central tunnel. The tunnel, this is the central tunnel consisting of two calcium ions. The upper calcium ion, which is green by color, that is the catalytic calcium, whereas the lowest calcium ion, that is red by color, that is the structural calcium. So the dissociation of the structural calcium leads to permanent denaturation of this enzyme. So calcium is very much essential for the activity of this enzyme. It's a calcium-dependent enzyme. So these are the various substrates of this human PON1. It has been categorized into aryl esterase, lactonases, and organophosphate. So the organophosphate has two other categories, that is organophosphate pesticides and non-agents. So particularly we are concerned with the paraoxon pesticides, since we aim to increase the activity of this enzyme by mutation. So first of all, we took a representative substrate, that is the paraoxon. After that, we tested the activity of all other substrates. So the neurotoxicity of these compounds. Acetylcholine esterase is an enzyme which is present in the synaptic cleft and between two neurons and in the neuromuscular junction. So the natural substrate of this enzyme is the acetylcholine, which is nothing but a neurotransmitter, and that is secreted from the axon terminal, from the vesicles, and it is taken up by the receptor in the next terminal end. So the extra acetylcholine neurotransmitter is degraded by the acetylcholine esterase enzyme to form the acetate and choline. So this is the natural process. So what happened? The organophosphates, that is the pesticides and nerve agents, they mimic the structure of these natural substrates, and thereby instead of binding the natural substrate to the acetylcholine esterase enzyme, the nerve agents, they go and bind to the serine residues of this enzyme, and they form a phosphonated complex, and therefore it entirely blocks the enzyme to and therefore more and more acetylcholine chemicals are deposited on the surface of this synaptic cleft, and that leads to the cholinergic crisis and the constant flow of information, because all these receptors are very busy, and constant flow of information goes through it. So the paraoxonase 1 enzyme that potentially degrades these nerve agents and pesticides, and therefore revive the entire process, and therefore this has been a drug candidate. In our lab, we have made a recombinant human PON1 wild type, which is around 99% similar with the human PON1. Okay, the only difference was that the recombinant human PON1 wild type was having an extra amino acid in the C-terminal region, and it was having 6 histidine tagged. That is for the easy purification through the nickel anti-chromatography. So this recombinant human PON1 wild type was subjected to site-directed mutagenesis to get this 7-point mutation, recombinant human PON1 7-point. So this 7-point mutation we took, that is by keeping in mind the fact that the recent molecules which was generated against the chimeric PON1 was having all these 7-point mutations, but it was chimeric PON1, and therefore we cannot deny the immunogenicity. So we have grafted all these 7-point mutation to our recombinant human PON1 wild type, and we did the site-directed mutagenesis studies. After that, we purified the enzymes, and later on we characterized the enzyme. with different panel of substrates. Likewise, in our lab also, we did site-directed mutagenesis study, the recombinant human 112P. That was, we did the site-directed mutagenesis study earlier, and in two positions, 115 position and 192 position, where histidine was replaced with tryptophan, and in 192 position, arginine was replaced with lysine. So it was having very good activity. After that, we decided to take this as a template for our next random mutagenesis study. And then we did the IV purification, inclusion body purification, and later we characterized the molecule. So the panel A that shows the linear recombinant human PON1 wild type, and this is flanked, the ORF with the green color ORF is flanked with the ND1 restriction sites and XHO1 restriction site, and the panel B that shows the digestion profile. Here we can see that at lane number 1, the pure plasmid is there. Lane number 2, it is digested by single digestion in presence of ND1, and the lane 3 that represents the double digestion. So here we can clearly see around 1.1 kb the ORF. So after that, in panel C and D, we can see this wild type mutant was subjected to site-directed mutagenesis to get the 7-point mutation. So after that, we checked the mutation by bidirectional sequencing, and we we concluded that all the sequence were present in their respective positions. So here we can, this is a multiple sequence alignment. Here we can see the naturally occurring PON1, wild type, and 7p. So the shaded area that represents the difference in amino acids in particular positions. So after that, we did the soluble purification of this enzyme. So it was done by 3 chromatographic procedures. First of all, we did the Q-separage, then the gel filtration chromatography, and finally we did the nickel-NT chromatography. So in the Q-separage, the panel A that represents the Q-separage, here the triangle, pink triangle that represents the paraoxonase profile, the activity profile of this enzyme, and the circle that represents the protein content, and the blue line that represents the gradient concentration of the sodium chloride, because we have eluted the sample with the gradient concentration of sodium chloride. After that, we did the gel filtration chromatography. The elutant were collected, they were pulled, and it was loaded onto gel filtration chromatography. And here from the gel filtration chromatography, we did finally the nickel-NT chromatography. In presence of 150 millimolar of imidazole, we eluted the sample. So the panel D and E that represents the SDS phase and the Western blot analysis of this molecule in different stages of the purification of this molecule. Here we can see that at lane number 4 in B panel, there is a clear band, there is a silver stain band, a clear band that represents the paraoxonase 1 sample. And from the Western blot also we clear that this is the paraoxonase 1 sample. So these are the different substrates we have taken for this study. First of all, we did the comparison of various hydrolytic activities against the paraoxone. Paraoxone is a synthetic substrate which is having paranitrophenol over here. So after degradation, paranitrophenol is deposited, and that gives a yellow color. So by that we can measure the yellow color at 405 nanometer. So here we can see that panel A, the wild type was having around 20-fold less activity in comparison to the recombinant human PON1 7p. 7p was around 20-fold more activity in comparison to the wild type. After that, we did the SEHE revival assay. In acetylcholine esterase assay, we saw, first of all, we incubated the enzyme in presence of different concentration of the organophosphate. After that, with a particular time point, in between, in equal interval of time, we elevated the sample and checked the SEHE revival assay. So here we can see that the data that were represented, here we can see that this blue color that shows the 7p, recombinant human PON1 7p, whereas the red color that shows the wild type. So the K observed was calculated. From the K observed, it was found that it was around 100 folds more potent in comparison to the wild type. After all that, we also checked the different other activities, hydrolytic activities like phenyl acetate, delta valorolactones, ester thiolactones, and the EHL hydrolyzing activity. So here we can see that in phenyl acetate activity, the 7p was having 1.8-fold less activity in comparison to the wild type. Whereas in homocysteine thiolectin and the delta valorolactam, these are two lactams where activity was more or less similar. The next we check the activity of 3-oxo-C12-EHL. It's a quorum sensing molecule, and since the PON1 that degrades the quorum sensing molecule, it's a quorum sensing quencher also. So here we can see that in comparison to the wild type, the recombinant human PON1 7p was around 6-fold more activity. After that, we did the random mutagenesis study. For that, we took the plasmid, that is the recombinant human PON1 2p, 2-point mutation 1. So this is the plasmid that carries the gene of recombinant human PON1 2p. So it was subjected to the transformation in the XL1 rat cells. It's a mutatus stem. So after that, we got the colonies. The colonies were scattered, and that was inoculated into 10 to 20 ml of media. After that, we got the mutation. The plasmid was isolated, and it was further transformed into BL21D3 cells. From BL21D3 cells, after transformation, we got different colonies, and all colonies were individually taken into 96 well plates, and in 96 well plate, induction was done, and finally cell lysis and the activity for the positive clones are checked. This is a representative chromatogram. Something problem is in the slide actually. So this is a representative chromatogram. Here we can see one bar that represents a specific clone, and here one panel that represents the 196 well plate. Here we have taken at least 8 samples, 8 colonies which are controlled. This green one that represents the control molecule. So out of 1400 colonies we screened, we got only one colony which is having the maximum activity, that has been shown as the red bar. So in the early screening process, we got around 9 different colonies, out of which 2 colonies were found to have a good activity, that is 892 and 784. This was just checked the colony. So after that we took 784, that is one of the colony we took, and we did the SDS patch and the Western blot analysis. After that, the antigenicity of this molecule was checked by Western blot analysis, and equal amount of protein were loaded. Equal amount of protein were checked for the paraoxonase-hydrolyzing activity. And here we can see that the wild type, the recombinant human PON1 7p, the recombinant human PON1, that was around 2-fold in comparison to the wild type. So next we did the random mutagenesis study using error-prone PCR method. See, the first generation mutant was had the plasmid carrying the first generation mutant, that is 784, was subjected to error-prone PCR, and the mutated PCR product, the ORF, was ligated into the digested vector backbone, and the ligation for that product was transformed into DS5 alpha cells. After that, according to that procedure which we earlier mentioned, we did the screening process, and the activity of positive clones were checked. So this is the random mutagenesis using error-prone PCR method. Here you can see that panel A that represents the digestion profile, panel B that is the amplification, and panel C that represents the SDS space, panel D that is the Western blot. So equal amount of proteins were taken from the Western blot analysis, and here we got the 4 different molecules. From 3 to 6, we got 4 different molecules. One that represents the template molecule, and 2 that represents the first generation PON1 molecule. So the paraoxonase-hydrolyzing activity of the most effective mutant was found to have around 8-fold more activity in the crude lysate. So this is the overall results of recombinant human PON1 2p mutagenesis. Here we can see this is the template molecule. In the first round of random evaluation, we got 137 position. At 137 position, allele was replaced with threonine. In the second round of evaluation, we got 4 different molecules which are having different mutations. After that, we check the inclusion body, we purified the inclusion body. So earlier, we had taken, earlier we had done the purification by soluble methods. Here we did the inclusion body purification method. So the inclusion body purification method was desirable. That is because in inclusion body purification method, around 90% of the protein are purified in a single step. Whereas in case of the soluble expression, it takes 3 or 4 different steps, particularly in paraoxonase 1, to get the purified protein. So, and also in our lab, we have already seen that in 1 liter of protein, 1 liter of cell culture, we got around 500 mg to 1 mg of protein. Sorry, 500 microgram to 1 mg of protein. Whereas from purification through the inclusion body purification, we got around 20 mg protein. So because of this yield, we decided to do the purification by the inclusion body method. So here you can see that this is the purification procedure, and the purity of protein at different stages of IV purification was represented with the SDS patch and the Western blot. So here we can see that this is the around the 40 kD band that represents the inclusion body, but some contaminants are always present in the purification procedures. So after that, we screen the inclusion body. We wanted to refold the inclusion body. For that reason, we screened the refolding conditions. So we have developed a scheme of 310 conditions having various kind of buffers and chemicals. These are the different chemicals we have taken for the screening procedures. So the screening process that includes, first of all, the inclusion body was solubilized in 8 molar of urea, and the concentration was taken into 1 mg per 50 ml of your refolding buffers, and after that it was incubated at 25 degrees centigrade in constant shaking, and after overnight incubation, the refolded protein we got. So we screened 310 different conditions, out of which 2 conditions were found promising, which is represented in red. So after purification, we separated. We took the Q-separase ion exchange chemotherapy to separate the active protein from the inactive molecule. So this is the purification profile, where we can see that 2 peaks are there. So the black chromatogram that represents the protein content, the peak 1, where we are getting the activity profile, the blue one that represents the activity profile, but here in peak number 2 we are not getting the activity. So that means some inactive proteins are there in the inclusion body, purified inclusion body. So after that, after Q-separase chromatography, we separated the inactive proteins. So from here also we can see that this peak 1 that represents the active protein, whereas peak 2 that represents the inactive proteins. So this is the SDS page picture of all refolded enzymes that we have made in the mutagenesis study. After that, we compared the soluble purified and the IV purified enzyme. The mutant protein RT460 was made through soluble purification as well as through IV purification, and after that we could able to see that there was no difference by purifying the enzyme by soluble method or by inclusion body method. So after that we characterize the enzyme. We did the kinetic characterizations. So taking the paraoxonase as the substrates. So here we calculated the V0 initial velocity. After that we plotted the V0 against the substrate concentration to get the Michaelis-Menten plot. So here so the Michaelis-Menten plot was fitted with hyperbola, 2 parameter equation. After that we got here the kcat by km values, where you can see that the RT460, which is the most active protein, was having around more than 30-fold greater activity in terms of kcat by km in comparison to the recombinant human hormone wild type. Whereas the 180 protein was around 14.8, that is around 15 times more in comparison to the recombinant human hormone wild type. So after that we did various other hydrolytic activities like the CPO and DAP. CPO is a pesticide, whereas DAP is a NORB agent. So here we can see that in the case of CPO, the RT41, RT460, that is the yellow color, that represents around 35-fold more the CPO hydrolyzing activity in comparison to the wild type, whereas the RT180 that exhibited around 21-folds more CPO hydrolyzing activity in comparison to the wild type. After that we also did the DFP, that is NORB agent hydrolyzing activity. Here also we found a very good activity. It was showing around 336-fold increase in DFP hydrolyzing activity in RT478. It was having around 336 folds increase in DFP hydrolyzing activity in 460, whereas in RT478 and 789 it was around 95%, that is 97 and 94 folds more activity. After that we also checked the different other hydrolytic activities like the delta valorolactones, 3-oxo-C10 homoserine lactone activities and thiolactone activities, but we found differential activity. However, in delta valorolactone activities and phenyl acetate activities, all the activities ceased due to our mutation. So this mutation is not favorable for the phenyl acetate or the delta valorolactones. However, we could able to get differential activity in the homocysteine thiolactone and your AHL hydrolyzing activity. So by this I would like to stop here. Thank you.
So I'm sure the speaker would like to answer questions from the audience. Maybe I'll start with one. You have used 3 different methods to induce to select or induce mutation.
One is XL1 red, another one is error-prone PCR method.
Right. Which one is the best, like in terms of?
Actually, it has its own limitation. XL1 red method, actually you see, whenever you are the desired gene that is in plasmid, whenever you are incorporating or transforming that gene, so what will happen? Other than the specific gene, other position will also mutate. So that is one of the your other person other than the ORF will also mutate.
Right.
And the error-prone PCR, it is very difficult to ligate the molecule because the ligation efficiency is very less.
Right, right.
But definitely error-prone PCR method is a very best method, what I have apprehended.
Mm-hmm. Now do you, after all this, do you select a few that you're going to go further?
Yeah, actually we have already patented this molecule, we have filed the patent. And actually we are also getting good signal from the companies also. So it is particularly useful for CW and a chemical warfare NORB agent. NORB agent are particularly used as a terrorist activities, particularly Soman and Sarin. You might have known Soman and Sarin, these NORB agents are particularly, that was used in Iraq war also. And it's a multifaceted enzyme. It can hydrolyze it can be used as a quorum quencher also. It can be used as the anti-atherosclerotic enzyme, and it can also be used as a what is that called? Like that we can use. It's a multifaceted enzyme.
Sounds good. Thank you.
Yeah. How about using bacterial phosphorylates? Because there are some bacteria which have the ability to degrade. The phosphate.
Right. Actually, there are 2 types of activities, particularly catalytic activities and stoichiometric activity. Some bacteria are there which degrade this molecule stoichiometrically. Means one molecule will hydrolyze. Only one molecule will be hydrolyzed. But this is catalytic scavenger. Means one molecule will catalytically hydrolyze a number of substrates. So this is particularly important.
That's good. Thank you very much.